this practice, we will use agarose gel electrophoresis to determine DNA fragment size and to quantify DNA extracted from blood cells and agastache mexicana, that is a plant. This technique separates DNA molecules based on size. DNA has a net negative charge that is proportional to its size. When placed in a matrix of agarose and exposed to an electric field, DNA molecules will move towards the positive electrode. The agarose gel is made of tiny holes. Smaller molecules move faster than larger molecules because they produce less friction in the agarose matrix.
Materials and equipment: agarose, DNA samples, DNA size standard, electrophoresis buffer, gel loading buffer, electrophoresis chamber, gel sealing tape, Erlenmeyer flask, trans illuminator, paper towels, microwave, power supply.
For this practice, the first step we need to do is seal the edges of chin, dry the glass of acrylic plate with tape to form a cowl. Set the mold on our horizontal section of the bench. The second step we need to do is to prepare the electrophoresis buffer, to fill the electrophoresis tank, and to cast the gel. The next step, we need to prepare 2 solutions of agarose in electrophoresis buffer at 0.8% and 1% concentrations, and we need to use a EM flask for each solution. The next step, the fourth step, is to heat the mixture in a microwave until the agarose dissolves, and we need to control this heat because we don't want to spill out the agarose if it boils. The next step, we need to use insulated gloves to remove the flask from the oven, and we need to pay attention to dry the agarose gel, so we need to add the ethidium bromide to the gel solution before it solidifies. But in this case, we didn't use ethidium bromide, so we use another type of acid to prevent the solidification. Once the agarose is dissolved, allow it to cool it down, and add 5 microliters of the solution we are working with per 100 milliliters of the gel to reach a final concentration of 0.5 microliters per milliliter. After that, we need to mix the gel with the solution by gentle swirling. The 8th step, while the agarose solution is cooling, we need to choose the appropriate comb for forming the sample slots in the gel position. Each comb is 0.5 till 1 millimeters above the plate so that a complete well is formed when the agarose is added to the mold.
Pour the warm agarose solution into the mold. Allow the gel to set completely, approximately 30 minutes at room temperature. Carefully remove the comb, trying not to break the gel.
Place the gel within the mold in the electrophoresis tank and add just enough buffer TAE1X to cover the gel to a depth of approximately 1 millimeter. Mix 4 microliters of your DNA sample with 1 microliter of loading buffer. Slowly load the sample mixture into the wells of the submerged gel using a micropipette. Place the gel within the mold in the electrophoresis tank and add just enough buffer TAE1X to cover the gel to a depth of approximately 1 millimeter. Slowly load the sample mixture into the wells of the submerged gel using a micropipette, glass capillary tube, or Pasteur pipette per sample. Load size standards into slots on both sides of the gel. Close the lid of the gel tank and attach the electrical leads. Apply a voltage of 100 volts for 30 to 45 minutes. Keep an eye on the loading dye migration as two colored bands should appear on the gel. Run the gel until the bromophenol blue and silane cyanol have migrated an appropriate distance through the gel. To stop the electrophoresis, turn off the electric current and remove the leads and lid from the tank. Visualize the gel using UV light in a trans illuminator and capture the gel image on the digital camera equipped with a UV emission filter. Send the gel photograph to your email account. Write down all your observations, and once you have printed the photograph, paste it on your lab book. Label the samples and the size of each fragment from the size standard. Compare your results from two different gels, 0.8% and 1% respectively, and describe the different factors that have affected the DNA sample's migration through the agarose.
After analyzing the run that we obtained in the electrophoresis, we found that there was no DNA in none of the 4 samples. After comparing our methodology with the other teams, we found out that the only difference was the electrophoresis apparatus. So we believe that our apparatus used a higher voltage due to a decalibration, causing our DNA sample to run faster than expected.
Electrophoresis gel is used in medical diagnosis and in molecular biology for know the size for different species, plants, bacteria, or in molecular diagnosis.